Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 67, and it's going live on July the 10th. Bit of a disaster this week, as the weather was finally compliant and we got some filming done. Only when I was downloading the files, my cat jumped on the laptop, and I have no idea how he managed to delete the whole lot. Even AI couldn't find them. Last week, our guests were Jeff Fisher, President of Longhorn Vaccines and Diagnostics, and Hannah Ames, Founder and CEO of Levita. It's certainly been a very popular episode. This week, our conversations are with Amy Walker, CEO of 4Base Bio, and Adam Tomassi-Russell, Senior Director, Climate at Deep Science Ventures. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or for event coverage, or to moan about FIFA. First, it's time to go through some of the headlines of the articles you may have missed in the past week over at deeptechdigest.com. A new tool is giving scientists a clearer view of DNA regulation. A new immunotherapy strategy targets aggressive brain tumors and their fuel, and human embryo gene correction research has been approved. SK Bioscience launched an initiative to reduce uncertainty in vaccine development decisions. 1023 Health has received Swiss medical licenses for its Basel and Visp facilities, and there are new approaches to the prevention and treatment of polyomaviruses. PFAs in medicine can often be replaced with alternatives. A new approach to rare disease drug development yields its first research results, and Novartis is acquiring Myrix Bio. We had an article on synthesizing DNA using only temperature. A new CRISPR method makes it possible to control protein production in cells, and you can read all of these and plenty more at deeptechdigest.com. All right, let's get to this week's interviews, and first we're talking to Deep Science Ventures, which is a venture creator building deep tech companies in 4 sectors: agriculture, climate, computation, and pharmaceuticals. And of course, it's that last one that we're interested in, as the company recently formed a partnership to work on crossing the blood-brain barrier. To tell us more is Adam Tomassi-Russell, Senior Director, Climate at Deep Science Ventures. Hi Adam, thanks so much for being with us today. I wonder if you could start by telling us a bit about the company.
Yeah, so Deep Science Ventures has been around for getting on for 10 years now, and the founders of DSV, Dom and Mark, spun DSV out of Imperial College London with an aim to think about how you might create impact and ventures in a very different way to the traditional technology transfer model. So DSV takes a very invention mindset approach to thinking about how we solve the world's biggest problems. And so what that really means for us is we start with a high level outcome that we ultimately want to solve for, which may be solving for various diseases in the case of health and therapeutics, through to climate problems and agricultural problems. And what we do is we try to understand what is ultimately holding back our ability as a society to solve those problems. And in this way, what we do is we unpick from that high level outcome in a first principles way the different things that we need to solve for as an organization to make that happen. And that means we're not starting with a particular idea of the technology we're going to use to solve that problem and trying to find its place in the world. But what we're trying to understand is what are the constraints there, and what existing or novel scientific and engineering knowledge do we need to look at and combine in order for us to solve this problem. And I think when you think about it that way, there's a huge amount of science and engineering that's been discovered over the last 100 years. And we have a bit of a special place in that we take that and use it and engineer it to solve these really, really important problems. We don't do that just on our own. So we apply this process with partners, but also something that's very important for us is, as we're ultimately looking at forming companies through this process of invention, that we find really, really high quality scientific and commercial founders which can join us on that journey and can ultimately work alongside us as co-founders to think about what it is we want to build in order to solve those problems. So I think we're very unique in that particular way.
I assume that those solutions differ depending on what aspect you're looking at.
Yeah, absolutely. What's really important to us is to be as impartial as possible. So when you're thinking about how to solve a problem, it can be very dangerous, I think, to zoom in too quickly, ultimately, on what the solution space looks like. When you step back and look at some of the startups that are working in various spaces, you can see that although they themselves have close competitors that are using similar solutions and similar technologies to what their company is doing, there is still also this plethora of other types of approaches which could actually deal with the high level outcomes. And so once you take that step back and say, actually, I'm not obsessed with how we do this, but I need to solve the problem, it opens up very different types of solution spaces across all the different outcomes that we want to solve for. And, over time, we've created as a business over 60 different companies which have solved problems in very different ways. It's also true when we build companies that we, as well as looking at different solutions that solve for these problem sets, that we also try and think about systems change in a sense of how can we build, in some cases, multiple companies which can in some way in the future interact and solve different parts of that puzzle, so that we look to build not only successful companies in their own right, which of course have to stand on their own two legs, but that we can also think about building a change in the way that some of these platforms are approached and how these things might work together going forwards as well.
And you mentioned the partnerships, and looking at your website and the information on your company, some of those are quite diverse, thinking of AbbVie, Cancer Research, Coca-Cola.
Yeah, absolutely. I think that speaks to sort of a few things, really. I think at the highest level, what it really speaks to is the diversity of problems that we as an organization try to tackle. Really, what's important to DSV is how do we create impact at the highest level, which can impact the human ability to thrive and for the planet's ability to thrive, and therefore allow us to have a thriving environment. And in order to do that, you end up with a hugely diverse set of outcomes you want to solve for. So on one hand, you're solving for disease that sits obviously within the body, but also you're looking at how do you think about making sure that we prevent disease through climate change and making sure that the environment is safe there, making sure that we have clean water that we can access and that we don't fall into water shortages, that we make sure that ultimately the ecosystem supports us, that agriculture makes sense for us. And so this is why we have such a wide array of different partners. And what's important to us really is for the partners we select or that we want to work with is that we really just care about solving for the problem, and that we don't, again, have any agenda in going into that we use particular solution spaces. And so we've been very fortunate over the last 10 years to find people that have sat either in a slightly more corporate ecosystem, in foundations and not-for-profits, as well as government entities that want to solve these problems. And for us, that's the most important thing.
I was reading an article the other day about water consumption by data centers, and the comments had descended into a bit of an argument where someone who clearly worked in a data center was saying how other industries used far more water, which of course sucked people in from those industries. And it just became a we're better than you are back and forth. How do you approach potential conflicts like that?
I don't necessarily see those always as conflicts. I think having the mindset that we're less bad than the other person isn't really a helpful way to look at this. In the end, what we have to think about is, we have to co-solve for a lot of these issues at the same time. And that's certainly what we do as an organization. So we're not looking at narrow-mindedly thinking about, well, we have to solve this particular agriculture problem, but we don't actually care about the externalities or side effects of solving that. I think the fact that we work across so many different spaces within the organization, from health through to climate, to agriculture, to computation, as one of the other areas we work on, that we're always mindful of constraining the solution space such that it doesn't affect these negative externalities, in a sense. So it's not to say, oh, we can solve for the problem with jet fuel, but we don't care if that ends up in a river. Like clearly these things need to be co-solved for, and that's something we do very intentionally and very explicitly. I think one of these things is just to be very mindful of these things as you're thinking about solving any given problem. And then, as you say, there's a this is better than this type argument, but the reality is when we're looking at what we want to solve for, we want to have the biggest impact we can. And so we work very hard to think about what are the opportunity areas that we want to work on, how big an impact can they have in their own right. And it's true that obviously we want to focus on the biggest impacts, but that doesn't mean we shouldn't be thinking about the impacts in a smaller way as well. So yeah, something we're very conscious of. And I think it's a good point because people do use those arguments.
Other company builders are available, but what is it that you do differently to some of your competitors?
Yeah, absolutely. I mean, we know a lot of the other guys that are building companies, and they're doing actually, a fantastic job. And our view is that the more people that are building to solve these global problems, the better. It's not going to be one person that solves all of this. I think the thing that we do differently, and I think how we fit into the ecosystem, is specifically around this impartiality to the solution space. And so by going through this first principle high level analysis, we want to make sure that we're not overly focusing on things that currently exist in the market. We don't want to focus on novelty necessarily. It's not always the case that novelty ends up in solving the problem. So I think that for us, it's that ability to sort of take a step back and not have any particular agenda in terms of how we go about solving those. I think that's sort of where we make the biggest difference in terms of what we do. As I say, I think there's lots of good reasons to use the technology transfer model. I think there are loads of successes there that have had huge impact. And so I just see us having a special place within that ecosystem and how we do it.
You've been around, as you said, for several years now. I wonder if you could tell me about some of the successes that you've had in that time.
Yeah, absolutely. So, we've been around for getting on for 10 years, and throughout that time, been interesting actually. We have, at least in the early years, tried a few different models to try and understand how we go about doing this. In the first instance, we trialed some of the more common models like challenge models where people come with solutions, but we found it often falls into the same trap. And this is why we do it the way we do. And over 60 companies now, we've had a lot of success. Our portfolio is now valued at more than $700 million right now. And people have raised hundreds of millions from really good investors, including Sequoia and Breakthrough Energy, Lower Carbon, Sam Altman. And we've had lots of companies making impacts. In terms of the work we've done in therapeutics specifically, I think there are a couple of particular standout examples just in terms of the stage they're at. And we've had a company called Mitos, who does automated cell manufacturing, raise a $19 million Series A. And we have a company called Antiverse who is focusing on using machine learning to design antibodies effectively. And they've raised more than $20 million in total as well. And although, as I say, we're impact focused as an organization, we do believe in many instances the venture capital pathway is the best way to scale. And we generally think about the successes of our companies raising this money as an indication of the ability to scale and have that impact.
Obviously, it's a life sciences podcast, so concentrating on that and talk about drug delivery, because one of the most challenging parts of that has often been targeting the brain. I wonder if you could talk a little bit about those challenges that you're working on.
Yeah, absolutely. So obviously, our partnership with MDC is focusing on the blood-brain barrier, which is itself well known to be really hard to penetrate. So the hardest thing there is how do you get the therapeutic interventions into the brain? And the blood-brain barrier is designed specifically to keep things out. It wants to keep out pathogens and toxins. And so it blocks more than, I think, 98% of small molecules and 99.9% of larger advanced therapeutics, biologics, and gene therapies. And so this creates like a major barrier ultimately to people being able to treat these diseases. And so a huge percentage of the world's population, 40%, live with some type of CNS disease, ranging from Alzheimer's to Parkinson's to various types of cancers. And the reality is our ability to actually drug those and to treat those diseases is blocked by our ability to deliver these effectively to the brain. It's certainly the case over time that people have delivered therapeutics to the brain, but they've generally been using suboptimal approaches to try and deal with those diseases. So people have had to use local delivery via transcranial surgery or intrathecal injection, or using other delivery vectors, or even targeting other drug targets around the periphery rather than going straight in for the CNS itself. So it's a huge opportunity and can make a big difference to a lot of people. And this is why we're focusing on this with MDC.
Has that become something of a barrier to companies getting into delivery of treatments to the brain?
Yeah, absolutely. From a venture perspective, I think historically people have been reticent to back therapeutics aimed at the brain for this reason. I think given the consistent increase in the need for these drugs, that people have been a little bit more willing to back some of these drugs that maybe aren't ideal, but can get through the blood-brain barrier into the brain. But these themselves are obviously non-ideal, and they often can have other effects that are non-desirable. And so it's certainly, from an investment perspective, challenging, but I think there's a huge amount of willing to solve this problem. And I think the market is showing that this is a wave of technologies that needs to be backed right now.
And obviously, you mentioned the partnership with MDC. Could you tell me a bit more about that and how that started and what you're going to be doing with them?
Yeah, absolutely. When we're looking to partner with organizations to build the ventures that we build, we want to find people that, one, really care about the problem, but two, can really add to our ability to solve that problem. And MDC is one of the UK's premier life science service companies for turning drug delivery concepts into these commercial realities. And they also have expertise in CNS biology, amongst other areas. And so in the same way that we look for partners normally, we're talking and engaging with key opinion leaders, we're engaging with scientists and commercial entities that want to solve this problem. This happened also with MDC, and obviously we both care about solving this problem. So what we're going to be doing going forward is working on this problem by recruiting in a founder in residence that has higher level of expertise in this area and high founder potential. And they're going to come in and work directly with us to think about what approaches can we take here to solve this problem, and how do we solve those high level outcomes that we want to solve for. So we're going to be looking for solutions that, for example, are systematically dosable, they're effective in adult BBB, they can get to the right places in the tissue, they can be used across multiple diseases, they have the right economics, low dosage frequency, all these things. So the idea is that we'll get that founder in residence in, and we'll spend this time exploring how do we solve for those with them. Once we get to a set of approaches we think are incredibly promising, what we do generally then is do further triaging to reduce this list down to one or two things we think are really, really promising. And we then bring in co-founders that have additional expertise that will complement the founder in residence. And we spend another 6 months with them, really nailing down all of the techno-economics, making sure we really understand the development pathway, the funding routes, and all these things. And so this is the type of thing we'll be doing with MDC on this project.
And will that involve refining existing delivery methods, or are you looking to new technologies, or is it going in with an open mind and seeing what works best?
Yeah, absolutely. I mean, clearly trying to solve this problem is incredibly complicated, but what we have to do is be impartial to that. And we're exploring a really wide array of different routes, through from novel specific transporters to looking at validated transporters, but augmenting them in some way to increase the performance, all the way through to synthetic transcytosis, amongst others. And so, really, we have a lot of space to explore here to solve the problem.
And so, how do you see that? Obviously, this is in its infancy in terms of the partnership, but How do you see this field evolving? And are you hoping that what you derive from this partnership will be applicable in other areas as well?
Yeah, absolutely. What we really want to target is that we can access as many CNS related diseases as we possibly can. But we really want to get to something where we can evolve the platform to, hyper specificity, hyper conditionality, making sure that whatever we do is increasingly well tolerated, administered as infrequently as possible, and overcoming some of these target mediated liabilities of the existing solutions, which limit the dosing window, etc. So, I think there's an awful lot to do here just within the current diseases and paradigms we understand today. And obviously, as the understanding of the brain increases over time, this will enable us even more so to think about other diseases that we might be able to use these delivery mechanisms for.
And as you move forward with some of the companies that hopefully will be working on this, what's your model? Are you a very hands-on company, or do you kind of let the experts get on with it? Or again, does that vary too?
It definitely varies for sure. The way we try and approach this really is how can we be as helpful as possible to the firms that we create? After we spin the company out from Deep Science Ventures, we stay involved in various ways. That can be being on the board of the company. It can not being on the board of the company. It doesn't matter to us too much. What really matters to us is that we build up a trusting relationship with our founders such that we're the people they pick the phone up to when they have a problem that they want to solve for, whether that be operational, technical, commercial. And so, yeah, we stay involved with the vast majority of our companies quite deeply, some more lightly. It depends exactly on the team and the founders' preferences. But we work with teams in earnest, I would say. Series A is our sweet spot in terms of where we feel we can make the biggest difference to the companies.
Some pretty big names for partnerships there. Next, we're headed to Cambridge, not the one in Ontario or the one in Massachusetts, but the one in the UK. 4Base Bio is a biotech company pioneering the use of synthetic DNA to enable next-generation therapeutics and vaccines. To tell us all about it is the company's CEO, Amy Walker. Well, I guess usually I just ask about the company, but you've had an interesting journey at the company, so I'm interested in your journey as much as the company itself.
Yeah, no, I have. I mean, I was the first employee at 4Base Bio when we spun out of our former parent company. So that's a little bit of a story in itself, actually. Our previous CEO ran a company called Expedion, which was more focused in the sort of tools and reagent space. And then it was sold off to Abcam at the back end of 2019, another UK biotech company. And at that point in time, we already had our DNA technology within Expedion, albeit that it was being deployed in sequencing, so for whole genome amplification and single cell amplification from biopsies. And at that time, we'd sort of recognized this unmet need, thinking about being able to deploy our enzymatic technology into large scale DNA manufacturing. And so we retained that technology. The rest of the assets were sold to Abcam. And from that point, we spun out 4Base Bio into its own entity in 2020. So the technology itself predates 4Base Bio as a company, but the first years were very, very interesting because we were well funded and we were able to just get our heads down and develop the technology itself. And when I say that we were working in microtiter plates, I mean, at that early stage of development, we were making nanograms of DNA. And to put that into context, today we're making grams of DNA at a GMP scale. So the first years were really, really fun. It was all about developing the IP and very scientifically rich and rewarding, building out the teams from scratch, which was, truly, truly a wonderful experience. And then in 2024, we sort of reached what we could call a value inflection point, and we wanted to see what we would do with the company. Would we sell the company? Would we keep going? We built out our own clean rooms for GMP manufacturing. And at that point, we took in a fairly sizable investment from M&G, the pension fund here in the UK, and also from Patient Square Capital, which is a US-based private equity firm, and they have been able to fund the company further to the completion of our GMP certificate, and really, the objective being the commercialization of our product and making sure our product is embedded into as many different programs as possible. So it has been a journey, yes.
And you've only been CEO for a few months now. How has that transition been?
That transition's been good. I think, I was incredibly fortunate because being the first employee, I've got to wear a lot of hats along the way. So most recently, I was serving as Chief Operating Officer, which I think gives me a good view of how the business runs. That was really my responsibility. And we kind of had this succession plan that I would be appointed as CEO. So it wasn't particularly shocking at the time. But of course, the job has changed. I would say that there's maybe a little bit less science than I'm accustomed to and a lot more investor relations, for example. But yes, it's been really great. And I think we've built out a really exceptional team to build the company forward.
But you were talking about that scale difference. I mean, in the years since you started as the first employee, there must have been huge changes.
Yeah, no, I mean, there really was. And I love reflecting on this because essentially the organization has had to expand to serve its purpose. So I could give a couple of examples for that. For the first couple of years of our development, naturally, we didn't have a quality team because we weren't making material for clinic. So we kind of had to build along the way. And in truth, we made mistakes along the way. We didn't really know what we were doing until we were doing it. And so watching the face of the business change, of course, the research and the development and all of that innovation is still part and parcel of 4Base Bio. I think we will always remain a scientifically driven and minded company. But really where the business has expanded is, of course, in quality manufacturing operations to make sure that we're able to make compliant material that is suitable for clinical trials ultimately is the goal. And then the most rapid area of growth over the last couple of years has really been the commercial side of the organization. So really scaling out that business when we were starting. Again, one of my multiple hats was commercial. So it was really a lot of that client engagement was done by me. And now I'm very pleased to say that we have an excellent chief commercial officer and an excellent team sitting under her that are intimately familiar with this sort of products that we sell and the markets that we sell into. And, I'm very encouraged by the results of that.
Related to the products that you sell, what are some of the challenges or limitations in the field right now?
That's a great question. And maybe I should have started with this. 4Base Bio manufactures DNA, but we manufacture it incredibly uniquely in that we use enzymes. And essentially the objective there is to displace an incumbent technology, which is a technology called plasmid DNA. Plasmid DNA is derived from bacterial fermentation. So it's grown in bugs, in E. coli. And as such, there are a lot of limitations associated with that manufacturing process. And I can talk to a couple of those. I think one is really the impurity profile. So being propagated in bugs means that there's a risk of host cell contaminants or endotoxins or any of these other contaminants coming from that bacterial fermentation. And in addition to that, you also need to use antibiotics. And so there's antibiotic resistance genes present in the final product, the final plasmid DNA. In addition to that, the lead times associated with getting something called GMP plasmid, which is plasmid that would be suitable for clinical trial applications and being dosed into patients. The lead times could be upwards of 6 months to get a batch. And the final point that I would make there is the kind of scale consideration. So in order to put that into perspective, to make 1 gram of plasmid or a couple of grams of plasmid, you might be in 2,000 liter bioreactors. And so the infrastructure and the scale of production and fermentation is an interesting consideration. So if we think about the product that 4Base Bio makes and making DNA using enzymes, the scale of manufacture is orders of magnitude smaller. So as such, our clean rooms are tiny. We can manufacture gram scales of this DNA in a couple of liters rather than thousands of liters. And so when you think about the scale out considerations, we're targeting either very large patient populations or N of 1 medicines being personalized cancer vaccines, for example. It is a rapidly scalable process, both upwards and downwards, and I think that's really important. In addition to that, the turnaround time is expedited. We can manufacture material that is suitable for the clinic in a number of weeks rather than months. And I think most importantly, at least from my perspective, is the safety profile. So by definition, our process is what we call cell-free. And so that negates any risk of any of those bacterial impurities being carried over into the ultimate drug substance. And because, this isn't a plasmid-derived product, we don't have any of that bacterial sequence present in our final DNA products. And so that allows our partners and our clients to reduce the doses required because they're only dosing relevant sequence rather than any of this off-target junk material, as we say. So I think the benefits are significant. To talk about the challenges that we might face in our industry, this is a new technology, and in spite of all of the limitations that I can spell out about plasmid, it is an incredibly established technology that is widely adopted and accepted by regulators on a global scale. And so really what we have to do is we have to work with our partners and we have to work with regulators to present why they can switch to synthetic DNA and what those benefits are. And I'm really encouraged by the feedback that we're getting, both from our partners, but importantly from the regulators, about how one would switch from this incumbent to something synthetic like us.
Could you maybe go into a bit more detail on synthetic DNA amplification technology?
Absolutely. So I mentioned already that we're using enzymes for our process. And what this really starts with is an amplification technique. So I mentioned in plasmids, the DNA is amplified by propagation. It is grown in these bugs, divides there. And instead, we're using a combination of enzymes to exponentially amplify our DNA template into something much more substantial. And so the way that we do that is really unique. So very interestingly and unique to 4base Bio, we own and manufacture our own enzymes that are used in the process. And to that end, we have a manufacturing facility out in Madrid in Spain that is exclusively responsible for the manufacture of these enzymes. Jim, you might ask, why Madrid in Spain? And there's a very simple answer. The concept of rolling circle amplification, which is the amplification technology that we deploy, was invented in Spain in the lab of Luis Blanco, who remains to be a scientific advisor to 4base Bio. And essentially that was the asset or the technology that was acquired into the previous company, the predecessor of 4base. And so we just had our little hub there. We have excellent technical knowledge base there. We have a lot of the former postdocs from the inventors of this technology. And so we have this little hub. And between you and I, Madrid is excellent for food, as we were talking about. So I really enjoy my visits out there. But back to the technology itself, we essentially deploy this rolling circle amplification to amplify up our DNA. And that uses two enzymes. One is an engineered version of an enzyme called Phe29, which is the DNA polymerase, except our enzyme has been engineered for fidelity. So if you think about the sequence context, it's really good at dealing with challenging sequences that are typical of cell and gene therapy more broadly. And then the other enzyme that we use in this manufacturing process is an enzyme called a primase. And essentially this primase is used instead of primers, which would be custom specific, which means we can control that reaction and it's kind of sequence independent. So independently of the clients or the partners that we're working with, we can deploy the same enzymes rather than having to design custom primers every time we receive a new sequence. So that's quite unique. I think the next unique part of our technology is actually that we can make different flavors of DNA. And I say flavors because I really mean flavors. So depending on the downstream application, people may want different configurations of the linear DNA products that we can make. And that means, for example, a viral vector client or a client that's deploying the technology for lenti or AAV production would want different features on their DNA than a partner that is using it for mRNA production, for example. And the way that we process our DNA allows us to configure the ends of the linear molecules to suit the downstream application. And it means that the products are very customized depending on who that user may be. So I think those are some really interesting USPs about 4base Bio's platform over some others.
What about scale? Is there an issue of producing at scale?
There's not. So, I mean, scale is an interesting one. I think enzymatic reactions have been scaled for decades, primarily in the food industry, actually. But if we look more adjacent to what 4base Bio does, we can look at the success of the mRNA vaccines for the COVID pandemic. And so those are enzymatic reactions, just like 4base Bio's, and therefore we know that we can control and scale that well. I think one of the things that allows us to do that is that our reactions are isothermal, which means that they run at one temperature, as compared to maybe competing technologies like polymerase chain reaction that most of these listeners might know from their times in the labs as an amplification method of DNA. That involves cycling of temperatures from as high as 95 to as low as, 27, 37. And so when you think about cycling temperatures at scale, that becomes an engineering nightmare, candidly. So I think it really benefits us that these are isothermal processes. And, internally, we've proven that we can scale up to multi-gram batches within our current facility. I think that the other interesting consideration is actually the complete other end of that scale. So if you think about this incumbent technology that I described, a lot of plasmid DNA providers are set up for really commercially scaled manufacturing that go into tens of thousands of patients or vaccines, whatever that downstream product may be. But with the advent and the success of some of these personalized cancer vaccines that we're seeing, or even personalized gene editing approaches like the baby KJ success, plasmid providers and CDMOs aren't really set up for scale out rather than scale up. And so the very neat thing about using an enzymatic process is that you simply reduce the volume and you reduce the single-use bioprocessing bag, and you can precisely tailor the batch size for the end user. And so that is very encouraging to me. And I think that will allow us to be much more agile in these emerging industries that really can be potentially curative for some of these patients. I think that, coupled with the express turnaround times that we can have against plasmid DNA, make this technology ideally suited for those sorts of patients.
Those are some of the advantages. Are there any other advantages of synthetic DNA?
Yeah, so I think that, we've covered the speed one, that's clear, and this kind of scalability on both sides of the spectrum. But for me, I think I'd like to reiterate the safety point. If we're thinking about these gene therapies and these therapies that are dosing patients, if you take, for example, the sorts of doses that are required for an AAV gene therapy, a systemic AAV gene therapy, be that DMD or SMA, you're thinking about dosing 10 to the 14 particles into these patients, into these pediatric patients. And if even 5% of those AAVs contain non-appropriate sequence, like we would typically see from plasmid, that's billions of particles being dosed that contain this non-target, non-functional AAV particle, which really should be a therapeutic. And I think 4base Bio, along with our partners, have done a really extensive amount of work to prove the safety of our synthetic DNA product. And people are starting to recognize that as a key benefit. So I think, to reiterate, there's definitely the safety benefits. There's cost benefits because our partners need to use less DNA than they would with plasmid. And certainly there's speed benefits as well.
You mentioned partnerships. Could you tell me about any of those and any of the results that you're allowed to share?
I can speak quite generally there. So, I think if you look at our client base and our partnerships, Jim, when we first started selling this product, and I like to tell this story, the only people that were willing to work with 4base Bio were crazy scientists, right? They thought the platform was super cool, very disruptive, and they were compelled by the science. And so we got to deal with all sorts of wacky sequences and projects at that point in time because of the clientele that we were engaging with. I am very encouraged by the fact that, yes, we still have that caliber of people within our client base, and they're very important because they're willing to take a bet and they'll partner with you and see you through to clinic. And, we're very impressed with that. But I think what's quite encouraging when we think of the broader adoption of synthetic DNA, historically, pharma partners are a little bit more conservative. And maybe a little bit more resistant to market trends than some others. And I think if you look at our client base today, we're working with 6 out of the top 10 pharma clients globally. One of those pharma clients is already in a phase 2 clinical trial with us. So I think this nod from maybe that more conservative area of the industry kind of resonates with us and suggests that really this is being more broadly adopted and worked into their sorts of pipelines as well. So that's very encouraging. I think the other interesting consideration that we're working on is how does 4base Bio partner with CDMOs? So for example, those CDMOs could be making the downstream drug substance, be that a lentivirus, be that an AAV, be that mRNA. And how do we partner with them to ensure that we're getting the most reach for our product through those partnerships and, watch this space as that develops over the next couple of months?
Is it changing or evolving, and how do you see the field changing in the future?
Yeah, no, I think that's a great question. So we want to be ahead of that curve, and really we want to make products that solve an unmet need to our downstream partners, those drug developers. And I think one of the very encouraging things and the things that I love most about being a DNA provider is that actually you get to feed into all of these modalities. So whether our partners are making mRNA vaccines or they're making personalized cancer vaccines or they're making in vivo CAR Ts, our DNA can be a building block in all of those different drug development opportunities. And so we're constantly looking for market trends and seeing who's developing what and trying to get ahead of that curve. I think specifically our DNA manufacturing process is super flexible, so we can quite quickly change the enzyme composition to make new flavors, like I mentioned earlier, of those DNAs. Be that a single-stranded molecule, which people might like for gene editing, be that a circular molecule. There's lots of different ways that we can modify our DNA to make sure that we're serving our partners.
Is there an awareness issue? And do you have to explain this to people, or do they get it?
I think it's changing. I'd like to say that it's changing. So really, it was very much an educational uphill battle to present the benefits of our product, certainly when we had very minimal data and less of that kind of clinical positioning, because we weren't being adopted in clinic in the same way that we are today. So there really was that education piece. And I think something that we have done really, really well is that from very early in our development, we set up what I call application validation teams. And so what that means is internally within 4Base Bio, we have a small group that is working on AAV, a small group that is working on mRNA, for example. And in the first instance, they actually were developing data packages using our product against the incumbent technology plasmid so that we could present scientific data, because science sells. They need to see the proof in the pudding. It's all very nice that we say, oh, this is wonderful, but let's look at the quality of those products. Let's look at the product that you make from this DNA. And I think that was incredibly helpful that we were able to present technical data rather than kind of marketing data at a lot of different conferences, which was really how we started to engage with our client base in the first instance. I think we're becoming more widely known now, and I think some of that clinical validation alongside our license from the MHRA, which is the UK health regulator, that allows us to make drug substances and critical starting material, it brings that credibility. But certainly there's rooms that I walk into that nobody has a clue who 4Base Bio are, and we really want to broaden that reach and that adoption.
Are there any other products that you have?
I think that we will think towards other nucleic acids. So for example, I mentioned already, a lot of our partners are making mRNA with the DNA that we provide. So the question would be, well, does 4Base Bio ultimately want to make the mRNA as well as the DNA? And I think we really are nucleic acid experts. So that is one thing that we would consider alongside our partnerships. But, at the heart of it, we are a DNA technology and company.
How do you see the future in the short and long term?
The goals are adoption. So we really believe that 4Base Bio's technology is well placed to displace plasmid. I don't think there's any application area where there's a credible suggestion that plasmid can work in a way that synthetic cannot. And as a scientist myself, I truly see these benefits. I think that plasmid DNA market, depending on who you speak to, is worth $3 billion in 2026. And so we want to disrupt that market, and we want to make sure that people are waking up and thinking, why on earth would we use plasmid when there's a safer and more effective alternative? And that really is the goal and the mission of the company. From a personal perspective, I want to democratize genetic medicines. I want to make sure that these are accessible to the masses, and that's something that drives me.
Are there other companies working in this field, or are you on your own?
We're not on our own. So there's another UK company, a company called Touchlight. So really we position both ourselves and Touchlight as the leaders in this space of amplification, enzymatic amplification. Touchlight have been around for much longer than 4Base Bio, since 2007. But we really think that we've caught up with them, over the last 6 years with respect to our clinical readiness, for example, and the infrastructure that we've built to serve our partners. Other than that, naturally, you'll always think about emerging players into this space, and we do keep an eye on that. But I think there's kind of adjacent technologies, so synthetic DNA fragment assemblies like a twist or an IDT, for example. But really in the category of making material that's suitable for the clinic, both with respect to quality and scale, it's ourselves and Touchlight leading that space.
And do you have any plans on kind of expanding beyond, you mentioning Madrid and the UK? Are there any expansion potential?
So as it stands today, we have really great capacity from our UK facilities. We've recently announced a move to a new headquarter facility here in the UK, which will increase our capacity and really allow us to consider those N of 1 medicines that I spoke about earlier. I think a natural next step for 4Base Bio would be to look for a footprint in the US. A lot of our clients remain to be in the US. There's a lot of innovation going on there. But right now, we have that capacity and we're happy with what we have here.
Another really enjoyable conversation, including the bit before the recording where we were talking about restaurants in Belfast. I'm not sure what it is about me and food. Anyway, 4Base Bio doing some great things and definitely a company to watch. And so that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.